my name is Ivana, as I mentioned, and I'm in McDevitt's lab. And today I'll be talking about basically strategies to engineer co-emergence in organoid models. And so, in order to build organoid models that faithfully recapitulate aspects of native tissue, you have to have the correct cell types, the correct ratios of the correct cell types, and you have to get them into the right organization. And historically, the way that the field has gone about doing this is through this mixing approach, where you take human pluripotent stem cells, differentiate them into the cell types you need, purify these, and then aggregate them. And while this approach really does give excellent control over the ratio of different cell types, and you get robust and reproducible formation of these aggregates, you don't get macro scale structure formation. So really, this method doesn't take advantage of cells' intrinsic ability to self-organize the way that they do in vivo in development. So another method that our lab has more recently been interested in is this method of co-emergence, where you have your pluripotent stem cells aggregated, and then after aggregation, you provide them with differentiation. And we found that with this method, you get spontaneous, well, not spontaneous, but you get co-emergence of different cell types that spontaneously organize and replicate some aspects of in vivo development. So you get these large-scale structures forming, and even though this is a better model than our mixing model in some ways, to study self-organization, you get control of tissue type properties and relatively poor reproducibility. The question is, how can we make these co-emergent models better? So in both cardiac and neural organoids, these are just two examples from our lab, you see that in the co-emergent process, you get a rich diversity of cell types that are occurring in the organoids. And even though you get these high-level structures, the organization is, as I mentioned, poorly controlled, and the structures don't necessarily always form when you do the experiments, or there's a lot of batch-to-batch variation between experiments and between organoids themselves. So in order to control emergence of structures, we really need to understand what initiates their formation. And this is an interesting question, because in these very early time points, I can't show with my, I don't think they see that, but in the early time points, you essentially start with a homogeneous aggregate of pluripotent stem cells, and then somehow symmetry breaking events arise. And even in these very early time points, you can see some semblance of patterns forming. And the question really becomes, can we create complex tissue models by mimicking these early developmental patterns? So developmental patterns actually precede tissue organization every step of embryonic development, and then also well into adulthood. And there's a wide range of different patterns that give cells spatiotemporal information for where to go and how to differentiate. So some dynamic patterns that are, for example, in this case, this is a zebrafish example, are signaling pulses and gradients thought to be important for elongation and a slew of other things. Also, this is just one example. There's also density and mechanical gradients, as we see in this example of the mouse gastrula, where density gradients are continuously changing basically over the course of gastrulation and evolution. And then finally, the one that I will be focusing on is mosaic patterning events. And in many cases, these patterns are actually, the more complicated patterns are the result of an initial mosaic patterning event, which is basically where a subpopulation of cells acquires different structural and phenotypic characteristics, which then allow it to initiate this cascade of self-organization. So, as As I mentioned, mosaic patterning can be defined as the emergence of a subpopulation with different structural or phenotypic characteristics. And this is caused by a population of cells having heterogeneous response to signal, and some population of these having a heightened response, which then leads to this bifurcation event that eventually initiates an endogenous cellular organization and differentiation program. And so attempts to, recapitulate mosaic patterning events have led multiple groups to engineer circuits, for example, to essentially cause symmetry breaking events to occur in vitro. So one prominent example from Wendell Lim's group is this example of the extrinsic engineering of a synthetic circuit using a SYN notch. And I know Sebastian touched on this briefly as well, but the idea is that you can cause, through cell-cell interactions, you can cause a subpopulation of cells that overexpress E-cadherin. This is happening in my mouse fibroblasts, and eventually cause organization of these cells. And this is a great proof of principle to show that mosaic patterning can occur through manipulation of cell-cell adhesions. But this is an engineered system, and we know, for example, that in pluripotent stem cells, they have these intrinsic gene circuits that define how they will organize. So one example from our lab, from Ashley Libby, who is a previous graduate student, takes advantage of these intrinsic cell systems, and she shows that when you mix wild type cells with cells that have E-cadherin, CDH1 knockdown, that these cells separate from each other and organize into these pockets. And what's very interesting to me about this is that if you take these pattern colonies and you expose them to different differentiation treatments, for example, dual SMAD inhibition for neuroectoderm or CHIR treatment for mesendoderm, the cells themselves are biased from being in a pattern. That is to say, on the left side for the neuroectoderm, you see that wild type cells exclusively acquire PAX6 expression, whereas on the right side and are more biased towards neuroectoderm, whereas on the right side you see if they undergo mesendoderm differentiation, the CDH1 cells will preferentially express mesodermin. And this is something that we see also in different types of patterning events. So the idea is to use this platform with cell tracking and modeling to essentially predict what inputs would be required to create these high order desirable structures. And we recently, a paper came out of our lab, also Ashley Libby and David, who's here with me right now, and in collaboration with the Delta Lab, who will be presenting next, to try to predict, for example, if you control which cell types and timing of induction, then you can essentially have more complicated patterns form. And the idea is to use this to eventually make even more complicated patterns and eventually transition it into 3D. So long term, we hope to use predictive modeling to control self-organizing cellular domains within our organoids. So with that, I'll take any questions.
So thank you for that. How, in your models, how deep do you get into, the biological mechanism and downstream pathways, any kind of feedback regulation and other elements that interact besides, that specific adherence?
Right. I think I didn't mention this, but I think that's definitely one thing that is precluding us from getting more complicated patterns, that we don't have a we haven't thoroughly characterized all the molecular regulators of self-organization. And we know for a fact that, for example, when you knock down certain proteins, there is compensatory mechanisms that step in and
sorry, to get to one, part of it is framework limited, I would argue, or I would say. So the modeling framework that we started with is one that can recapitulate the features of the physical, mechanical sort of interactions of the cells. Obviously, to go to where I think you're saying, Ron, to add that other layer of feedback. So in past works with like Melissa Kemp, part of it was the rules-based ones, but then to start to introduce, instead of probabilistic distribution or probabilistic density functions, to start to be able over time substitute those with the actual molecular regulators. So we're not there yet, but that's the type of intent that we're going to. So that basically, yeah, you could have that, but you know, as you I think know well, that requires a different type of modeling because you've got to integrate multiple frameworks with different dimensionalities to them, both spatially and temporally.
Nobody else is asking. All right, I'll chime in with another question. And this is, we're kind of having a debate about this in the lab right now. So, you know, you have the cadherin effects early on. So as the cells, you know, begin to sort themselves, cadherin seems to be playing an important role. And we've also, you know, done that in other contexts with CHO cells, for example. But the person who did this work, Jessie Tordoff in our lab, she thinks that once this kind of coalesce into these bigger spheres, then the cadherin effect is going to be. She thinks that, you know, because I've been trying to convince her to do like a later cadherin experiment with iPSCs going into organoids, and she thinks that by the time you get to like, you know, 5, 6, 7 days into it, cadherins are going to have no effect whatsoever in sorting. I don't know what your thoughts about that are.
Which cadherins, I guess, would be my first question. So
we're E, N, P.
Yeah, so there are changes with all of those that we've seen in some of our different models, E, N, and P. There's actually VECAD in other work, not as much in these because it lacks a lot of the vascular ones. So whether they are responsible for sorting events or associated with sorting events, that's the kind of thing that you can't really easily tease out, I guess. We know that from these, that you predispose, you get the organizational. It's actually with the system we're using, kind of one maybe benefit in retrospect is ECAD is only transiently silenced. So it comes actually back on in some of these systems because as the CRISPR-I machinery in this particular construct gets silenced, that we think epigenetically, then what happens is you regain the ability of the cells, of some of the cells at least, to be able to express E-cadherin. So it's really just setting up the early divergent events and then letting the system sort of go back almost to a fully operational state. So it depends, I think, on how you would induce it, but if they're not sorting if they would not sort and organize, like in our structures with these where there's demonstrable changes in phenotype and sorting events, we see occur over the course of like 2 weeks. If it's not cadherin mediated, then I don't know what else would be driving it. That's what's driving most of the developmental ones. There is perhaps some matrix, but not of the nature that the cells really would migrate on in the type of 2D sense. Motility in a 3D one, Emily and I were talking about this yesterday. Motility is different in a 3D sense like this. It's a collective sort of function. And so again, I think a lot of the 2D rules that people are used to applying just aren't directly relevant. It's also one of the reasons then that I think you can extrapolate some of the properties like for modeling purposes. But the more that you can get the measures in your in situ system, the better off you are for your modeling. It's more accurate.
Okay, great, thank you.
Now, when it's chosen hex, I don't know. Those are vastly different with the regulation mechanism. So
I've been trying to convince her to do it in iPSCs, and that's where I've been getting kind of pushback. Any questions? Kaylee, should we move on?
Certainly. We can move on to the last talk in the series, which is Newton.
Ivana, thank you for the talk.
Thank you.